This is Unit 3, Part D, Mitochondria and Metabolism. We'll look at a little background first, then briefly at building material, and finally spend a fair amount of time looking at energy. Metabolism is the total chemistry of a cell. Cells are composed of organic compounds, matter. Cells require energy in a usable form. Two types of metabolism are anabolism, the building of molecules, and catabolism, the breakdown of molecules. All metabolic reactions require enzymes. Metabolism provides two things: building material, the molecules that organisms are made of, and energy, mostly in the form of ATP, needed to carry out all of its functions. Let's look at building material. Now, the diagram looks rather complex, and we'll use it several times. Building material includes the molecules of life, carbohydrates, lipids, proteins, and some others. Generally, any substance can be transformed into any other substance. For example, glucose can be changed to amino acids. Fats can be transformed into sugars. Proteins can be changed into sucrose. Energy is available mostly in the form of ATP. ATP is the major form of energy used by cells. We think of it as a primary energy carrier. What is ATP? Well, it stands for adenosine triphosphate. It's composed of adenosine, which is closely related to adenine, one of the nucleotides, and three phosphate groups. When the third phosphate group is removed, energy is made available for the multiple uses by the cell. The ADP, the adenosine diphosphate that results, can pick up energy in a phosphate group and regenerate the ATP. To understand where ATP comes from, we need to answer four questions. What is the role of oxidation? What are coenzymes and what is their role? What is cellular respiration? And what is fermentation? Let's look first at the role of oxidation. Oxidations release energy that comes from any oxidizable substance. Usually, it's the form of an organic compound. We call that food. It requires oxidation-reduction reactions, and both of these have to occur together. In an oxidation, energy is always released by the substance being oxidized. Electrons are always released by the substance. Remember, electrons can be thought of as packages of energy. Hydrogen is usually released by the substance because hydrogen carries an electron with it. Oxygen, on the other hand, is sometimes gained in an oxidation. In a reduction, energy is always gained by the substance being reduced. Electrons are always gained because, again, these are packages of energy. Hydrogen is usually gained because they carry the electron with them, and oxygen is sometimes released. Let's look at a few details of oxidation-reduction reactions. An oxidation and a reduction must happen together. The substance being oxidized is a reducing agent necessary for the other substance to be reduced. The substance being reduced is an oxidizing agent necessary for the other to be oxidized. The reactions were originally named because it was found that when something was oxidized, it gained oxygen. Like the other details were discovered later, it often involves oxidation and reduction of permanent molecules. Coenzymes, for example, usually in solution in the cell. They release or take hydrogen. NAD and NADH2 are examples. They generally generate roughly 3 ATPs each. You'll see about this later. FAD and FADH2 generally produce 2 ATPs each. Many coenzymes are supplied by vitamins. Cytochromes are another type of permanent molecule. These are attached to the inner membrane of mitochondria, and they release and take energy in the form of electrons. Which is in the reduced state, NAD or NADH2? Well, remember, when something becomes reduced, they gain hydrogen, and so the NADH2 is in the reduced state. What is the role of the reduced coenzymes? They carry energy from processes that oxidize organic compounds, such as the Krebs cycle, to the electron transport system. What is cellular respiration? First of all, we need to look at the mitochondria. These are organelles that generate energy. Think of them as the power plant of the cell. A mitochondrion is enclosed by a double membrane consisting of a smooth outer membrane and a folded inner membrane. The folds are called cristae. Cellular respiration occurs mainly inside. Mitochondria are self-replicating. They contain DNA with genes that produce transfer RNA, ribosomal RNA, and mitochondrial proteins. Cellular respiration is a process that oxidizes organic compounds to generate energy. It is aerobic, that is, it requires oxygen. Glucose is the major source, but other organic compounds can also be used to generate energy, such as fats and proteins. We use glucose in our discussion because it's a useful frame of reference, and it's the most common source of energy for the process. Cellular respiration generates up to 38 ATPs per glucose molecule. It can be summarized with this reaction. C6H12O6, that's glucose, plus O2, oxygen, gives CO2, carbon dioxide, plus water, and of course, energy. It has 3 parts. We'll discuss each of these 3 parts separately. The first is glycolysis. It doesn't generate much ATP, 2 per glucose molecule. It occurs in the cytoplasm of the cell. The second is the Krebs cycle. This occurs inside the mitochondria. And the third is the electron transport system, ETS, also inside the mitochondria. Notice the black dots on the cristae. Those are components of the electron transport system. Many of the reactions in respiration are oxidations, in which NADs are reduced to NADH2s, FADs are reduced to FADH2s. We'll look at these 3 steps, as I mentioned, in order. Glycolysis. We can define it as the breakdown of glucose to pyruvic acid. It occurs, as we said, in the cytoplasm of the cell. Each arrow in this scheme usually represents many reactions. Now, it looks complex, I know, but it's actually a quite simplified look at cellular respiration. Glycolysis begins with one glucose molecule. Glucose is a 6-carbon molecule. Usually it exists as a ring-shaped structure, but for convenience, I'm stretching it out into a straight chain. 2 ATPs are required to activate the glucose. The first step is to activate the glucose to form glucose 6-phosphate. The ATP gives up one of its phosphates to the glucose. It joins at carbon number 6, leaving ADP. The glucose 6-phosphate is a rather important substance. It's an important crossroads substance because it can be transformed into glycogen or starch or sucrose or other sugars, and they in turn can be changed back into glucose 6-phosphate. A reaction with 2 arrowheads represents a reversible reaction, or at least a reversible process. By the way, this idea of activation to get energy out of the glucose is not really a foreign idea to you. Take a bucket of gasoline. It's loaded with energy. How do you get the energy out of the gasoline? Well, you have to activate it. It's easy to do. Activation means you add a little energy to get a lot of energy from the substance. What do you do with gasoline? Strike a match and drop it in the bucket of gasoline. The second ATP supplies a phosphate to the glucose 6-phosphate to produce another 6-carbon compound. This has 2 phosphate groups attached, and it's called glucose 1,6-diphosphate. The 6-carbon compound is split into 2 3-carbon compounds, and then a slight modification takes place to produce 2 compounds called PGAL, phosphoglyceraldehyde or glyceraldehyde 3-phosphate. PGALs then undergo several reactions to become pyruvic acids. 2 NADs are reduced to 2 NADH2s. 4 ATPs are generated. 2 of those ATPs were required to activate the glucose. So since 2 were added and 4 are released, the net is 2 ATPs generated from glycolysis because 2 of them were to pay back the system. The 6-carbon glucose has become 2 3-carbon pyruvic acids. After glycolysis, the 2 pyruvic acids are transported into the mitochondria, oxidized to 2 2-carbon acetyl-CoAs, and carbon dioxide is released. That's the 3rd carbon of each pyruvic acid. 2 NADs, meanwhile, are reduced to 2 NADH2s. Remember, for every oxidation, there has to be a reduction. Let's summarize the process. Beginning with one glucose molecule, an ATP is used to activate the glucose. It gives up a phosphate and becomes ADP and forms glucose 6-phosphate. Another activation step occurs in which ATP releases a phosphate, forming ADP, and the glucose 6-phosphate has become a 6-carbon compound with 2 phosphate groups. That's the glucose 1,6-diphosphate. Several reactions then take place, splitting that into 2 PGALs. Again, several reactions occur in which NAD is reduced to NADH2, 2 phosphates are added, and the result is 2 3-carbon compounds, each having 2 phosphate groups. 4 ADPs then accept 4 phosphate groups to become 4 ATPs, leaving pyruvic acid. 2 NADs then are reduced to NADH2 as the pyruvic acid is oxidized to acetyl-CoA. Notice also that the pyruvic acid released carbon in the form of carbon dioxide because the acetyl-CoA has 2 carbon atoms each. The second part of cellular respiration is the Krebs cycle. We can define it as the oxidation of acetyl-CoA. And the reason for that is most of the reactions in the Krebs cycle are oxidations because they produce a lot of reduced coenzymes. The reduced coenzymes, remember, pick up energy from one place, in this case the Krebs cycle, and transfer the energy to another place, the electron transport system. Let's begin with acetyl-CoA. This is another important crossroads substance because it can be generated from carbohydrates, lipids, or proteins. It combines with a 4-carbon compound to form a 6-carbon compound. This happens to be citric acid. In fact, the Krebs cycle is often referred to as the citric acid cycle. Citric acid undergoes several changes until it becomes isocitrate, another 6-carbon compound. The isocitrate is oxidized into a 5-carbon compound called alpha-ketoglutarate. At the same time, the NAD is reduced to NADH2. It's a decarboxylation reaction in which one carbon is removed in the form of carbon dioxide. Then a 5-carbon compound is split into a 4-carbon compound. This undergoes a series of reactions, mostly oxidations, that lead to the 4-carbon compound we began this process with. In each Krebs cycle, one FAD is reduced to one FADH2, 3 NADs are reduced to 3 NADH2s, 1 ATP is generated, and 2 carbon dioxides are generated. Now, don't let the lower diagram confuse you when you look at the upper diagram. We say 2 carbon dioxides in the lower diagram, and yet there are 4 in the upper. Well, that's because the upper diagram shows 2 Krebs cycles, which resulted from 1 glucose. Where does the carbon come from that's found in the organic compounds of the Krebs cycle? It came from the food you ate, maybe a sandwich you ate yesterday. What happens to the carbon dioxide that's released from the Krebs cycle? Well, it diffuses out of the cell into the blood, it's transported to the lungs, and it's exhaled. Right now, you are exhaling a waste product from cellular metabolism into the room where you're located. What about the third part of cellular respiration, the electron transport system? This is an ATP generator. Electrons and hydrogen are supplied by reduced coenzymes NADH2 and FADH2. The electron transport system is composed of a chain of cytochromes, abbreviated CYT, plus various proteins located on the inner membrane of the mitochondria. Cytochromes are alternatingly oxidized and reduced, and in so doing, they pass electrons from one end of the system to the other. For example, when the first cytochrome is in the reduced state, it becomes oxidized, giving up its electrons to the second. When the second acquires the electrons, it is reduced, and it turns right around and releases the electrons to the third cytochrome. The second has been oxidized, and the third has been reduced. And this continues to the end of the system. As the electrons are passed down the chain, energy is made available to generate ATP from ADP and phosphate. Again, it's the most efficient ATP generating system. It's not the only way ATP is produced. It's produced by a process called oxidative phosphorylation, because a lot of oxidations take place. It needs a continual source of electrons and hydrogen. These are supplied by the coenzymes NADH2 and FADH2. Each NADH2 will generate 3 ATPs. Each FADH2 will generate roughly 2 ATPs. The electron transport system requires a final electron acceptor. Final electron acceptor is abundant, easily reduced, and harmless when it's reduced. Guess what it is? The answer, it's oxygen. The final electron acceptor accepts electrons and hydrogen and is reduced to an end product. Ideally, the end product must be harmless or even useful. Guess what it is? Water. Some animals, like a desert rat that lives in the Arizona deserts, never drinks water, or rarely, and eats food that has a low water content. Most of the rats' water comes from the tail end of the electron transport system. Let's look at some details on how ATP is generated. The inner membrane of the mitochondria contains a complex enzyme called ATP synthase. Here's the space between the two membranes, so this is the outer membrane of the mitochondria. Hydrogen is pumped through the inner membrane into the space and then diffuses back through the ATP synthase, and in so doing, it provides enough energy to link phosphate to ADP to generate ATP. To help review the process, let's see if we can figure out how many molecules of ATP would be generated from one molecule of glucose. Glycolysis would generate how many ATP molecules? 2. Now you may think, oh, 4? No, 2 of those are used to pay back the 2 that the cell provided to activate the glucose. How many NADH2s would glycolysis generate? 2 in these processes and 2 more in these processes, a total of 4. How many reduced coenzymes and ATPs would 2 Krebs cycles generate? The 2 Krebs cycles would generate 2 FADH2s, 6 NADH2s, and 2 ATP molecules. How many total ATPs are produced by cellular respiration? Cellular respiration would produce 38 ATPs from each glucose. 4 come directly from glycolysis and the Krebs cycle. That's the substrate level phosphorylation. The electron transport system transforms NADH2 into 30 ATPs. Each produces 3, and there are 10 NADH2s. Likewise, the FADH2 would result in 4 ATPs being produced. Although the total of 38 ATPs theoretically may be produced, the cell uses some for various processes, and so less result for use by the cell. Some ATP is needed to transport the pyruvic acid into the mitochondria, for example. Let's look at the other form of metabolism to generate energy, fermentation. If you look at the diagram, it looks mighty familiar. Fermentation is anaerobic, so it occurs under conditions of low oxygen. It also can generate some ATP pretty rapidly. It involves the breakdown of 1 glucose to 2 pyruvic acid molecules. Instead of progressing into the Krebs cycle, though, the pyruvic acid is transformed into an end product, lactic acid. This is common in your muscles for quick bursts of energy. In yeast, instead of lactic acid, they produce ethyl alcohol as an end product. And once upon a time, humans discovered that if you mix yeast with fruit and fruit juices, it ends up with a delightful beverage. Can you imagine if you had a mutation and your lactic acid gene was changed to an ethyl alcohol gene? Well, when you did a little exercise, you'd get drunk. I guess it's a cheap way to have a party. Can you imagine among wild animals? If a lion produced ethyl alcohol and it chased after some food down the plains, it would collapse in a drunken state, or it got too far. Well, of course, the antelope would also collapse in a drunken state before it traveled too far. So that wraps up the cell. You're ready now to progress to Unit 4, cell division.